My name is Ingrid Mejia, and I'll be presenting tight junctions. So cells have cell-to-cell junctions that help cells join in order to form tissues and other parts. One of these means in order to join together are tight junctions. Tight junctions are used to seal cells tightly to form tissues and create a barrier in order to prevent diffusion of molecules, fluids, or ions. The barrier created is selective, and molecules that are not able to cross require proteins. So the figure on the left shows the structure of tight junctions and also some of its main proteins involved, which are occludins, claudins, and GEMs. Occludins help tight junctions and the efficiency of barriers and its stabilization. Claudins help that with interlocking with one another in order to form a tight junction between cells. And GEMs, or junctional adhesion molecules, are used when joining endothelial cells and leukocytes together. They're also used for polarization of the cells. There's also scaffolding proteins, that are zonula occludins, which interact directly with occludins and claudins. Tight junctions are found in cells such as epithelial and endothelial cells. The usage of tight junctions in urinary bladders is to make sure urine stays in the bladder. Also, their usage in endothelial cells is to create a selective barrier between the blood and the brain. Here are some of the diseases that are caused by the disruption of tight junctions. For example, the Crohn's disease, it includes broken barriers because of tight junction malfunction and epithelial apoptosis. Another example is celiac disease, where the disruption of tight junction causes intake of gliadin into the mucosa, which is in the first phase of the disease. Many of the disruptions on these type of junctions is caused because of its proteins. Either they're inactive or there's a mutation or a change on the proteins. In an article about a recent finding, the researchers were asking if Claudin 12 was an important type of protein in the blood-brain barrier and wanted to know the role in that which it played. They used a mouse model and inserted a lag-Z cassette in the reading frame of the protein in order to investigate the function of the protein. Claudin 12 was expressed in various cells, and which were also in the brain, but when it came to its role under non-inflammatory or neuroinflammatory conditions, it did not show an essential role. Also, because of the low numbers of Claudin 12 in the brain, they finally concluded that it did not affect the blood-brain barrier and it wasn't really required. And my question about the topic was, are tight junctions involved in wounds or injuries? And if yes, what is their role? And this concludes my presentation. Thank you.